Welcome back to part 3 of our macromolecules screencast. Today we're going to learn about nucleic acids. So we're familiar hopefully with nucleic acids. The N and the A in the molecule DNA stands for nucleic acids. Nucleic acids, we'll tie them real quick. They have a definite connection with proteins. We talked about in the last screencast about how primary structure of proteins is determined by this sequence of amino acids bonded together. And really the directions to how we bond those together all is found in the genes which are found in your DNA. The DNA directs the production and the lining up of all those and the connecting together of all those amino acids, and we'll talk more about that later. A little bit about DNA, what it does, a couple of things. First of all, DNA kind of provides directions for its own replication. We'll talk about that shortly. And it also directs RNA synthesis, which then in turn is going to control protein synthesis. So real quickly, if you look up here at this picture, we start over here with DNA, and DNA will make a copy of itself, complementary pair up one side of that double strand and make a strand of RNA. We call that messenger RNA. That messenger RNA will snake out and kind of leave the nucleus, and it will come out here and it will attach to this molecule right here, which is called a ribosome. And the ribosome is the site of protein synthesis. And at this ribosome, what that ribosome will do is it will read that messenger RNA 3 letters at a time and bring in the correct amino acid that correctly matches up to that 3-letter codons, what we call those, in that messenger RNA. We'll learn a lot more about that later. This process, by the way, up here is called transcription. We transcribe the DNA into RNA, and then this process here is translation. We translate that code into an actual sequence of amino acids, which makes that polypeptide, which eventually will make that protein. So we'll learn a lot more about that later on. So the basic sequence of things is DNA to RNA to protein. The next thing I want to talk about with DNA is how a little bit about its structure. So we talked about monomers and polymers. So the monomer in a nucleic acid are these little molecules. Oops, let me back that back up here. Are these little molecules called nucleotides right here? Nucleotide is the monomer of a nucleic acid. And it consists of 3 parts. First of all, it has a phosphate group right here. That's a little circle. You usually represent it by a little circle here. You see the circle there, and one there, and one there, and there. And then that's going to be attached to this sugar molecule, this pentose sugar. And those are going to make up what I like to call the backbone of that DNA right here. So they make up kind of the backbone, the supporting structure of that DNA. So if you think of it as a ladder, it makes up the sides of the ladder, and it's just there for support. There's no information, there's no genes found in that phosphate and that sugar backbone. Just provides support. All the information where the genes are in these nitrogenous bases here. Sometimes we call them nitrogen containing bases. And they're going to be the rungs of the ladder, and there's where all the information is. There is, you know, the letters, which you may have heard about, the A's and the T's and the C's and the G's are all those nitrogenous bases. In fact, there's three types of nitrogenous bases that we call pyrimidines: cytosine, thymine, and uracil. They left a U out of there, so that should be a U right there. Uracil. Now, real quick, we'll mention that uracil is only going to be found in RNA, and thymine is only going to be found in DNA. It's one of the differences between DNA and RNA is uracil and thymine. Down here in the purines, we have adenine and guanine. An easy way to remember which is a little saying that to help your little mnemonic device to help you remember these is pi cut. That is going to be the parent pyrimidines are cytosine, uracil, and thymine, pi cut, and the purines, you remember it by this little saying puag, is so the purines are going to be adenine and guanine. you'll be surprised how well that sticks with you. You'll remember that for months and probably years to come if you need to know the difference between those two. But again, remember that when we're bonding them together, that the A's always bond with T's, or U, I guess I should put in there, and C's always going to bond with G's. So that's a key role that we need to remember about DNA and RNA. Real quick, a little bit about DNA replication. When cells reproduce, okay, when a cell reproduces, there's one cell, and it's going to undergo a process called mitosis, in which it splits into two new cells. Now, before that can happen, that cell needs to ensure that the two new cells have the exact same DNA in them that was in the original cell. And so this process, DNA replication, has to take place before mitosis and before a cell can split into those two cells. So what happens here under this process is you have the original DNA strand over here. We'll notice in blue you have this original strand here. We'll trace it down here. What happens is that DNA will uncoil and unwind, kind of unzips. There are proteins involved, and when it does that, this side here serves as a template for a new strand of DNA. So some nucleotides will come up here and pair up with the complementary base pairing, will pair up with that old strand, forming a new strand of DNA, and they bond in there together like that. Okay, and then this other side over here does the same thing. We can trace it down here, and it's going to come out, and it's going to uncoil and unzip at the same time this other one does. And then again, nucleotides will come in and pair up with that old strand, forming a new strand of DNA. And what you end up with is two new strands that are exactly identical to each other, and they're exactly identical to that original strand of DNA. And so now all that DNA, we have two copies of it, so that they can both go into the two new cells that are going to be made through mitosis. We will talk more about that later as well. The last thing I want to talk about is this process that we mentioned in class called antiparallel. Let me get my pen over here. Let me spell that out. Antiparallel. Now, when we think of things as being parallel, we think of it like that. So this antiparallel doesn't mean perpendicular. Antiparallel, I like to think of it as like this. So the two strands of DNA, they kind of run opposite of each other. So real quick, and we won't spend a lot of time because we're about out of time here, we have the 5' end and the 3' end. You notice how they run opposite the 3' end and the 5' end over here. Real quick, the way we get those numbers is based on the carbon. So this is carbon number 1 right there, carbon number 2, carbon number 3, carbon number 4, and then right there, that little bend is carbon number 5. So that makes this the 5' end on that strand of DNA. And down here, so that's carbon 1, this is carbon 2, there's carbon 3, and it's on the bottom, so this is the 3' end. Same way over here, this is carbon 1, carbon 2, carbon 3, making that the 3' end. And down here we have carbon 1, 2, 3, 4, and right there is carbon number 5. That's all goes back to organic chemistry and the naming of molecules and how we number those carbons, the continuous long string of carbons there. But the main thing you need to remember is if they always run kind of opposite of each other. And here again, you'll notice in those nitrogenous bases, if we have an adenine over here, we're going to have a thymine over here in DNA. If you have a guanine over here, you're going to have a cytosine over here. So the G's and C's and the A's and T's will always match up with each other. That's enough on DNA that we want to talk about on your nucleic acids. So that wraps up the macromolecules. Hopefully this was some help for you. Again, we'll talk a lot more about DNA later on in the year, and you'll probably learn a lot more than you really wanted to learn about it. But anyway, thank you very much. Goodbye.